Log on, tune in, find out. Another good idea from Cambridge.
Microbial communities by high-throughput sequencing.
Thank you. This is ongoing work jointly with Michael Dufoin-Platel and Wally Gilks at Rothamsted Research. And Rothamsted Research is one of the oldest agricultural experimental research centers here in the UK. And one of the things that we have there, amongst other things, is the oldest experiment on permanent grassland in the world. It was established back in 1856, and its original purpose was to measure responses under different treatments of the plots, so in terms of plant diversity. So here in this slide you can see an aerial view of the experimental field, and you can perhaps distinguish, well, how the different plots look different because they have been treated differently. So up to recently, what people at Rothamsted had been measuring was rather in terms of things going on above ground. But in the last few years, technology has allowed us to access the DNA of soil of these microbial or bacterial populations. And we are talking about really complex communities. So this has brought several new questions, answers, and challenges. And here I am just including a few of them. So for example, we want to know which taxonomic groups populate the soil and in which proportions, what type of genes, functions, or pathways are present, and how those community composition affect essential life cycles and plant health. And when I say that this is a challenge, it's because biologists believe that there are as many as 100 million of different bacterial cells in this much of agricultural soil. So this is really a soup of many different things, and we are trying to make sense of what populates this much soil. So, and this is relevant because recently a group of several different countries have joined efforts in an initiative to sequence or parkgrass, this experimental field, to take it as a reference soil for other sites or from which fields treated differently can be compared. And this will lead to understanding on how to better manage agricultural soils and how to minimize negative impact of practices. It also should lead to understanding on how climate change is affecting these bacterial communities. So what we actually, the data that we observe and we are working with comes from next generation sequencing technologies, in particular 454 technology, and what we end up with is an incredibly complex soup of very short DNA sequences that originate from all across the genome and all across the different organisms that populate soil. And what we try to do is to tell something about the identity of these short reads, metagenomic reads, and if possible tell something about their evolutionary relationships with known things or between themselves. I think I didn't mention it before, and I think it's very important. 99% of the things that live in here have never been studied or isolated in lab. So that's why we are dealing with something, well, challenging. So one of the ways people go about this is to take this soup of metagenomic reads and compare them with known sequences available in databases, generally through the BLAST algorithm, for example. So in this slide, the sizes of the nodes correspond to the number of metagenomic reads that have hit that particular reference sequence. So here we get an idea of how this community is composed, and we get also an idea of what are the most abundant species in that community. But the problem with our particular data sets is that, again, most of the things are unknown. So these are just examples of results that we obtain by applying that similarity-based approach. And you can see here that the really large gray slice in this chart corresponds to unknown things. So the algorithm cannot really tell what they are. So we want to be able to tell something about all those unknown reads. And a different approach, not ours, I mean this was introduced by someone else, is to take a biologically well-established reference phylogenetic tree that is inferred from available reference sequences in databases. And so you take your another element that you need for this method is a marker gene. So again, you have reference sequences, but just for one marker gene. And this gene has to meet certain criteria. So it must be universal, at least across, in our case, all bacteria. It must exist in one single copy, because if we want to make statements about diversity and abundance, we don't want to count twice. So it needs to be single copy gene, and it must be not amenable to horizontal transfer. So once we have this reference marker gene, we take our soup of metagenomic reads, and again we identify the reads that align to that marker gene, and then we go on and place them into the reference phylogenetic tree. And once again, you can see that we are able to, the blobs, so here the red blobs correspond to reads that hit that particular edge in the reference tree. So this edge seems to get loads of hits. So we can say that our data set contains lots of reads that seem to be closely related to this clade in the reference tree. But again, this blob corresponds to reads that hit that edge, but we are not really obtaining any information so far about the evolutionary relationship between the reads or their exact identity. We just know that they belong to this part of the tree. So we want to see if we can go further than this, and so we reconstruct the phylogeny of the reads that hit that. We take a blob of interest in this reference tree and reconstruct the phylogeny of the reads that hit at that part of the tree. And then you can see that once we obtain this phylogeny, we are able to tell something about how diverse my reads are at that particular gene and at that particular blob of the tree, and also about how the different sizes of my clades, for example, what is the dominant clade, and etc. But all this is based on one gene, and we expect that there will be some sampling error on that gene, so we cannot rely on making generalizations just based on one gene. So we actually do the same and take a second gene, reconstruct the tree for the reads, and here it comes our problem. How can we align or find the correspondence between these two trees so that we can make statements about abundance and diversity of my metagenomic community. And this is, of course, very well known and studied for labeled trees, but here we are dealing with partially or totally unlabeled trees. So how do I find the correspondence between these two trees? And just to illustrate how we go on about this, these are my two trees. I arbitrarily take an internal node. It's 4 children branches, and you will see that I will collapse all the children branches that bend down from that tree. I will collapse them in a big fat node of size proportional to the number of the size of the clade. So I do that for the whole tree, and I do the same for the second tree, and then I ask what is the best matching between these two things. And you can see that the abundance of things bending from one edge is represented by this node. So if I can make this tree align according to their abundance and perhaps according to their branch length, then I will be able to find an agreement between them. And so this is the algorithm that we follow. So we make some distributional assumptions on the branch length of the trees, and then we test for the null hypothesis that says, well, this branch corresponds to this other branch in the tree, and we want to see how much evidence the data set contains for that matching. And we do the same in terms of the abundances at the tips. We make the distributional assumption of having a binomial distribution on abundances, and we test for this null hypothesis. So just graphically, that means if I have two trees and I want to match them in terms of their branch lengths and abundances at the tips, and I propose this matching between the two of them, so these two edges match and the two sub trees or clades to one and the other side match in this way, I would get a low score for this case because their abundances and also branch lengths don't seem to match really well. If I attempt this matching, I get a very low score, but then if I match them in this way, I get a high score. So perhaps this is the best way of matching the trees. And so just to show you how this algorithm actually works, I have created a simulation. So consider that this is the true tree that relates our metagenomic reads, and here there are the abundances. So you can again consider that there are some branches that have been collapsed into this big fat node, and so that means that there are very few branches bending from this node and from this point downwards. So you can, if you observe this, you could tell, well, this is the clades represent the diversity in my community. The nodes represent the abundance, but also I have then a big clade with high abundance, a large clade with very low abundance, and so on. So I want to see if the algorithm is able to retrieve that truth. So consider that these are the two trees that I obtained from my two marker genes, from two marker genes. Of course, we don't expect that the gene trees will have the same tree topology, there will be some discrepancies in tree topology, and because they are a sample, hence the abundance just as a sample at the tips of the tree. So if I match these trees, I obtain something like this, which is far from the truth. But then if I follow a greedy algorithm in which I then input this resulting tree and I take a third gene to match them in between, then I obtain something that is looking closer to the truth. Again, I apply the algorithm again. So I take a fourth gene and I match the two input trees, and there you go. So it seems that at least the information on abundance and size of clades is being recovered. So I would be, of course, we don't do this only 4 times. We have a set of 31 marker genes that meet these conditions that I told you about, and so that means that we would be, we do it, at least we match between 31 genes. In a different simulation, I wanted to look at the role or importance that branch lengths and abundances at the tips have in this algorithm. And to do that, I generated a tree and I matched it with itself, but without any information of abundance at the tips in this case. So you can see that because it's a tree with itself, so here I am measuring the accuracy of the algorithm. So I obtain 100% accuracy and the matching is perfect. But as I perturb the branch lengths of the second tree and I try to match them, then my accuracy decreases. And whenever the 2 trees that I try to match are very different in terms of the branch lengths, my accuracy really drops. But once I have trees with abundance information at the tips, then again I start by matching 2 identical trees, identical in terms of branch lengths and abundance. I obtain 100% accuracy. I then perturb the branch lengths and my accuracy doesn't drop as dramatically as before. So you can see that the abundances at the tip are really driving this matching process. So to summarize, at Rothamsted we are interested in microbial communities for a range of reasons. We have designed an algorithm for making statements about abundance and diversity in bacterial communities that compares to totally unlabeled phylogenetic trees and finds a consensus between them. We are, at present, we are still running more sensitivity analysis, and we are implementing the algorithm in an R package. And we are also planning to work on the quantifying the uncertainty in this matching. Thank you very much to our colleagues at Rothamsted and the BBSRC for funding and the organizers of this workshop. And I would like to finish by announcing a meeting that we will be hosting on behalf of the International Biometric Society next October on the statistical analysis of metagenomic data. And please visit our website for more information or contact us. Thank you very much for listening.
This is a technical question. When you have your reference tree and then you're saying you find edges that get many hits. What do you actually do to decide it gets many hits? Is that an MCMC chain with a fixed tree, and what do you actually do to? calculated?
Yes, well, this is actually work done by von Merig, a group of people in Germany, in the European Molecular Biology Lab, I believe. And as far as I know, their method has a probability distribution on each node of the tree, and that way, so for example, which you can is similar to the Felsenstein pruning algorithm, and given that probability, you can calculate if a read corresponds to that node or that branch of the tree, and that's how you assign. In specific, they do it by maximum likelihood.
Another question over here.
So in the analysis of metagenomic data, you end up aligning your breeds to not necessarily to leaves of the phylogenetic tree. So you could be using a classification tree, and I assume that you do. But then I don't see why you at the end get rid of all this information and treat the trees as unlabeled.
Yes, yeah, that is right, because I guess because we want to make statements about the unknowns, and to do that, you need to zoom into the blob, the node, which is not a node properly, the blob that contains all those hits, and try to say something about them.
What reference tree were you using?
Well, in this case, there are a few options, but it could be trees reconstructed from all the fully sequenced genomes currently available online, or it could be, I think we have also tried a tree constructed with the concatenation of our 31 marker genes.
So the tree you take from the literature, is that the Ciccarelli et al. Tree, or?
Oh, sorry, I didn't.
You said the Ciccarelli et al. Tree from 2006.
Ah, yes. Well, no, we are not taking specifically that tree. We are taking the list of marker genes from their paper,
Okay.
which is what subsequently von Merig used also as the marker genes.
The front here.
What would you do if you have some reads which align to many places and some reads which don't align?
In which method? Do you mean reads that align to the tree?
Reads can align to many reads to tree. What would you do?
Yeah, it depends on the method we are talking about, because if we talk about similarity-based placement of reads, then actually Daniel Hussen's group has a method in which you then don't place the reads on the tips of the taxonomy, but you place it to the most recent common ancestor of all the tips that the read hit. So you place the read in an internal node, but that is a similarity-based method.
You can, even on one genomic sequence, your read can be placed in different places. So,
Yes.
Could you repeat the question? So the question was, what happens if you have a reed that hits many tips of the tree?
in
Yeah.
many places on the same genomic
Yes.
sequence.
It depends what we are talking about.
You can hit many places on the same genomic sequence.
And or hit many places on the same sequence?
I see. Yeah, that okay. I can see your question. I haven't done the bioinformatic cleaning or processing of the data. It is interesting, and unfortunately, Michael is not here. I don't know what he does. You mean when you filter at this stage where you filter the reads for one marker gene? Is this your question?
You have also reads from, not necessarily for marker genes, you have all kind the mixture, so you don't know where your reads come from. You want to get reads from this 31 marker genes, but you have all kinds of.
Yes. Yeah, there are different ways of. So Michael will be here on Wednesday, and we can perhaps talk to you about that.
I think we might leave that question there, so you guys can maybe discuss it.
Yes.
We have time for one more.
Okay.
Thank you.
Very quick question.
I also read Van Mering's paper, and if I remember correctly, he concatenated the alignment of the 31 marker genes to reconstruct to get a better to match them against the reference tree. You choose a different approach by looking at the genes separately. Why did you do this?
Well, looking at them separately, but then trying to find a consensus amongst
exactly.
all them.
Yeah.
Again, because we want to make statements about the unknown. So, von Merig got to this point. Actually, this is a kind of output that von Merig has. Because we want to look into these blobs. So we want, because the reads that hit here could come from or have a phylogenetic structure on themselves. So we want to look into them.
All that short. Thanks again for a very interesting.